So, hi everybody. My name is Raz, and first I want to thank the organizers for giving me the opportunity to present my work here that I am working on in Andy Dillon's lab at UC Berkeley. So I'm interested in aging, and we know that there is a close link between aging and mitochondrial dysfunction. And what I'm going to show you here today is that improving mitochondrial homeostasis in a very small number of cells in an animal can actually reverse some of those aging phenotypes. So there is a major drive to try and maintain homeostasis in the complex eukaryotic cell. And the function of the cell is governed by the function of proteins. So not surprisingly, several protective pathways have evolved that protect the proteins in the cell and drive their folding and respond to different perturbations. These pathways are under siege, especially in the context of aging, where the cells are accumulating more and more damage, including protein stress. So there is more and more protein stress, and the activity of the protective pathways declines. Now, each different compartment in the cell actually has a specialized pathway that protects it. And I'm most interested in the mitochondria, which is, of course, famous for its role in oxidative phosphorylation and creating energy. And interestingly, this organelle also is very susceptible to protein stress due to its oxidative phosphorylation role. Previous work from the Dillon lab showed that triggering mitochondrial stress, specifically in neurons in the nematode C. elegans, actually resulted in a lifespan extension. And when they triggered mitochondrial stress in neurons, they actually induced a protective cellular pathway that protects mitochondria called the unfolded protein response of the mitochondria, or the mito-UPR. And this is a transcriptional program that includes antioxidants and mitochondrial protein chaperones and many other genes that are meant to alleviate the stress. Next, they wanted to go one step deeper into this and try and understand this on the cellular level. And they found that neurons are actually communicating with other tissues in the organism, such as the intestine, and upregulating this protective response in those tissues. So this led to the hypothesis that neurons are actually regulating longevity via a secreted signal. And this coordination was important for the lifespan extension. But is this really the whole story? Are neurons really the most upstream cellular regulators of this phenomena? So the nervous system is composed from more than just neurons. And there's an understudied population of cells in the nervous system called glia. And these are cells that do not conduct nerve impulses, but instead they support, nourish, and protect the neurons with many different functions that are emerging in recent years. And C. elegans actually provide us with a great opportunity to study these cells since it has a simplified nervous system. So C. elegans have 302 neurons, which account for 30% of all the cells. And they have 56 glial cells, which have been shown to be similar to vertebrate glia by morphological and functional criteria. So we wanted to ask, can glial cell extend lifespan via mitochondrial stress responses? So to do this, we trigger this mitochondrial stress response in different subsets of glia using a tissue-specific promoter driving a positive regulator that is sufficient to induce the mitochondrial UPR. And when we did this, we found that activating the mitochondrial UPR in different subsets of glia or in most glia was able to extend lifespan. And we were most struck by these CEP sheath glia or cephalic sheath glia, since these are only 4 cells in the animal. So what are these cells? These are 4 cells that are found in the head of the worm, and they ensheath the nerve ring of the worm, which is the major neuropil. And they have been shown to be most similar to the mammalian astrocytes. So next, we wanted to see whether these glial cells can actually communicate with other tissues similar to neurons. So we used a fluorescent reporter where GFP is regulated by the promoter of the mitochondrial protein chaperone. And we're looking here at worms that are lined up next to each other. So here are their heads and here are their tails. And here is the fluorescent image where the fluorescence is very low. But then when we trigger the mitochondrial stress in glia, which are here in the head, we actually get activation also in other tissues. We also quantify this using large particle flow cytometry. So triggering the mitochondrial stress in CEP sheath glia caused a lifespan extension on the organism level. And on the cellular level, it caused activation of this response also in the intestine. And for us, a very interesting question was, how is this communication happening? So what is the secreted signal between these tissues? And luckily for us, we have knowledge from previous works in the lab where we learned about how neurons are doing this interaction with the intestine. And these works found that serotonin, a Wnt ligand called AGL20, and the release of dense core vesicles from neurons was required for the signaling between neurons and the intestine. So we wanted to test these regulators. And in general, we wanted to test secretion pathways. So we wanted to test secretion pathways for neuropeptides, which is governed by dense core vesicles, and for neurotransmitters, which is governed by the release of small clear vesicles. So we muted each one of those genes. And we found that mutating neuropeptide processing, the release of small clear vesicles, dense core vesicles, or serotonin was able to reduce the induction of the mito-UPR in the intestine. So they were required for the signaling. However, all of these are mutations in all of the cells of the organism. So we can decipher whether the effect is coming from one tissue or the other. So to tackle this, we use the conditional knockout system where we can knock out the gene either in glia or in neurons. So when we did this, and this is to cut the long story short, we saw that glia cells only utilize small clear vesicles in order to release the signal, while neurons are utilizing only dense core vesicles to release the signal. And this suggested to us that there is actually some linear process here where glia signal through small clear vesicles to neurons, and then neurons utilize those previously described mechanisms through dense core vesicles to communicate with distal tissues such as the intestine. So now we can add this to our model. So these cephalic sheath glia use small clear vesicles to release the signal to neurons. And we were interested to find whether neurons are just bystanders and relaying the signal to the periphery, or whether do themselves actually change in response to the signal. And remember, we're talking here about protein homeostasis, and protein homeostasis in neurons is very important, specifically because of neurodegenerative diseases, where protein aggregation is actually the underlying molecular trigger in many neurodegenerative diseases. So we wanted to ask whether these glial cells can affect protein aggregation in neurons via this cell communication. And to test this, we use the Huntington disease model, where the neurons are expressing a polyglutamine tract, which aggregates in neurons and causes neuronal dysfunction. So we first tested for protein aggregation. So we use the filter trap assay, which is a assay where the aggregates stick to a membrane and you can measure them with an antibody. And we saw that activating the mitochondrial stress in glia was able to reduce protein aggregation in neurons. And we also saw that this reduction was dependent on the activity of small clear vesicles. And finally, we also tested for neuronal function. So worms can normally sense and chemotax towards specific odors, but when they have this polyglutamine tract in neurons, this ability is reduced. And we saw that when triggering the mitochondrial stress response in glia, we were able to partially rescue this defect. So now we can add this to our model where triggering the mito-UPR in glia causes secretion of small clear vesicles that are actually reducing protein aggregation in neurons. So putting this all together, we started by asking, can glial cells regulate longevity via mitochondrial stress responses? We saw that there are 4 astrocyte-like glial cells that can extend lifespan through this mito-UPR. We also did a lot of characterization of the cellular changes that are occurring in distal tissues, which I did not show you today. But we also see a lot of metabolic rewiring happening in the intestinal cells of worms. And what I did focus on today was the signaling pathway, the cellular signaling pathway, where we found that glial cells are utilizing small clear vesicles to communicate with neurons, and then neurons relay the signal to the periphery. And finally, I showed you that glial cells have an important role in neurodegeneration, where they can actually improve protein homeostasis in a Huntington's disease model. So I would like to thank my PI, MD Dylan, my mentor, Rio Higuchi Sanabria, and everybody that worked with me on this project, directly or indirectly. And thank you for listening.